Welcome all to the today's session. Today we continue with our embryology module. We are going to discuss second week of development, and then we will discuss about the third week of development. We start with our code master MRCOG with concept oriented clarity. This topic is going to be very important from the examination point of view. You will have at least 7 to 8 SBS from the general embryology module. Okay, so in the first week, what we have seen up till now was a fertilization, cleavage, and implantation begins. Okay, so let us recapitulate what we have learned in the last session. So what we have seen is the formation of the blastocyst and starting of the implantation. Okay, so on day 0, there is a fertilization in the ampulla of the fallopian tube. Then there is a formation of zygote, which is diploid in nature, will have 46 chromosomes. Then the cleavage begins. Okay, so you will have 2-cell stage, 4-cell stage, 8-cell stage. Okay, morula is a 16-cell stage. It forms by day 3. Okay, and it enters into the uterine cavity. Whenever you are reading about and learning about embryology, please see the timing. At what time what is happening? Okay, so there are questions on the various stages like morula 16 cell stage, it is formed by day 3. Okay, so you should remember these timings also. Then next step, morula gets converted into the blastocyst. Okay, so this blastocyst typically has got an inner cell mass which is comprised of embryoblast that will give rise to embryo proper, and then there is an outer cell mass. These are the cells which are surrounding. They are called as trophoblast, and that will lead to the formation of the placenta. Okay, then further what happens? There is a zona pellucida which is lost. That is the hatching, and then implantation begins around day 6, day 7. The trophoblast will differentiate into cytotrophoblast and the syncytiotrophoblast. Okay, so this is just to recapitulate what we have learned in our first session. Now coming to the highlights of the today's session. So what we are going to see is the second week, that is day 8 to day 14 after the fertilization. Okay, so the most important what is going to happen, the implantation will, which has started in the first week, will get completed. Then the bilaminar germ disc will get formed. Okay, so what is the key thing? It is a week of twos. Okay, so remember 2 2 2. What is this 2 that we are going to see? So implantation is completed by around day 10 to day 12. Bilaminar embryonic germ disc will form, and you will get 2 layers of the cells. So what are the 2 important cells? One is a epiblast and second is a hypoblast. So week of 2. The first 2 is a 2 cells, 2 types of cells, that is the epiblast and the hypoblast. Okay. Then you have 2 cavities. Okay. So the formation of the cavity. One is an amniotic cavity which lies above the epiblast, and second is yolk sac. It is from the hypoblast. Okay. So you have amniotic cavity and the yolk sac. The next is the extraembryonic mesoderm develops. Okay, so this extraembryonic mesoderm develops. The chorionic cavity will get formed. The development of the connecting stalk, that is the future umbilical cord, will be formed, and there will be formation of the primary chorionic villi. The syncytiotrophoblast produces SCG, and there is an establishment of the early uteroplacental circulation. So these are the changes which are happening. Okay, so uh, what are the two? One is an epiblast and hypoblast. These are the 2 types of cells. Then you have 2 cavities, amniotic cavity and the yolk sac. And third, there are 2 types of extraembryonic mesoderm which are getting formed. One is a there are 2 cytotrophoblast and the syncytiotrophoblast that is getting formed. So these are the changes which are happening, okay, in the second week of development. Okay, now let us see one by one. Whatever we have seen up till now highlights that we'll go into the detail. Okay, so in the third week, what is going to happen? That is the gastrulation and the organ systems. So second week highlights we have seen. Now we'll finish the highlights of the third week. That is the gastrulation and the organ system foundation. So here key theme is the trilaminar germ disc and the body axis formation. So in the second week, it was a bilaminar germ disc. Here it is a trilaminar germ disc and the body axis formation. So in the highlights, what happens in the week 3? There is a gastrulation. There is a formation of the primitive sticks. There is a formation of the trilaminar germ disc, that is ectoderm, mesoderm, and the endoderm. There is a formation of the notochord. This notochord will define the midline of the embryo. Okay. Then there is a beginning of the neurulation, that is by the neural plate formation, and then further there will be neural groove, neural uh, uh, plate will be formed and neural groove will be formed. Okay. Then the mesoderm, which is there, that will get differentiated into the three layers. That is paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. Then somite will begin to form. An early cardiovascular system develops, the blood islands and the primitive heart tubes, and the secondary chorionic villi will develop. Okay, so these are the changes which are happening in the second and third week. So first we will go into the details of the second week of development, then we will go into the details of the third week of the development. So highlights of the second week, we have just mentioned what happens. Now let us see how this happens. So what happens on the 8th day? On the 8th day after the fertilization, the blastocyst is partially embedded in the endometrial stroma. So see, this is the endometrial lining. This is the these are the glands of the endometrium, and this is the blastocyst. Okay, so you see two different layers. The one which is lined by the this tall columnar cells and blue in color, and second is a yellow cells. Okay, so this is the bilaminar, and there is a one cavity also you can see in the these are the epiblast above. So you have the amniotic cavity above, and you will have the hypoblast. So these are the epiblast and these are the hypoblast. Okay, so these are the epiblast, then you have the hypoblast, and there is a amniotic cavity, and this whole is an blastocyst, okay, which is getting buried inside the endometrium. Okay, so on 8th day after fertilization, the blastocyst is partially embedded in the endometrial stroma. The trophoblast differentiation, the trophoblast differentiates into two distinct layers. Okay, so this is another two which is happening, the cytotrophoblast and the syncytiotrophoblast. The trophoblast is of two types. One is a cytotrophoblast and another is a syncytiotrophoblast. The cytotrophoblast is the inner layer. It is composed of mononucleated cells. Cells have the distinct cell membranes and they are mitotically active. This is where the cell division happens. Remember, there is a difference between the cytotrophoblast and the syncytiotrophoblast. Okay, so this one differentiation is that these are the mitotically active cells, while the syncytiotrophoblast, these are the inactive cells as far as mitotic divisions are concerned. This is the inner layer, that is cytotrophoblast. It is a mononucleated, while syncytiotrophoblast is a multinucleated. Okay, these have a distinct cell borders, and syncytiotrophoblast do not have distinct cell borders. These are mitotically active. They are the syncytiotrophoblast. They are not mitotically active. So syncytiotrophoblast, that is the outer layer, multinucleated mass. There are no distinct cell boundaries. They are actively invading the endometrial tissue, but they are not mitotically active. So this is what is the difference between the cytotrophoblast and the syncytiotrophoblast. Okay. So cells in the inner cell mass. Now, last time we have seen that this blastocyst has got two things. One is a trophoblast and second is an inner cell mass. The trophoblast will divide into cytotrophoblast and the syncytiotrophoblast. Then this inner cell mass will also have two important things. The cells in the inner cell mass or the embryoblast differentiate into two layers. The hypoblast layer of small cuboidal cells adjacent to the blastocyst cavity. Okay, so these yellow cells, they are hypoblast cells. Okay, they are close to the blastocyst cavity. And then these blue cells, okay, epiblast layer, a layer of tall columnar or high columnar cells adjacent to the amniotic cavity. These are the epiblast layer. Okay, together these layers form the flat disc, which is a bilaminar germ disc. Okay, so there is an amniotic cavity. A small cavity appears within the epiblast. Okay, so amnioblast are epiblast cells adjacent to the cytotrophoblast. So these are the amnioblasts which are adjacent. This is covered by the cytotrophoblast. Okay, this whole is covered by the cytotrophoblast. So these cells which are adjacent to the cytotrophoblast, they are called as epiblast. Okay. So let us see the same thing here. Okay, so now you can appreciate it in a better way. So this yellow, these are flat cells. These are hypoblast. These are the tall columnar cells. These are called as epiblast. Okay, and then there is a cavity above. This is the amniotic sac, and this is the cavity which is below. That is this is the yolk sac cavity. Okay, so this is the primitive yolk sac. What we call it as a primitive yolk sac. Yolk sac also we will go into the details. So remember, first it is a primitive yolk sac, then it becomes the secondary yolk sac, then it becomes the definitive or the tertiary yolk sac. Okay, so this is the epiblast, this is the hypoblast, this is your amniotic cavity, and this is the primitive yolk sac. Okay, and this is the your endometrium. The implantation is complete. Here there is a fibrin coagulum which is formed. Okay, so and there is another layer which happens from this proliferation of the hypoblast is called as user's membrane. Okay, so this membrane is coming from the proliferation of these cells. Okay, this is the inner lining. This is called as user's membrane. Okay, so this yolk sac now is lined by this user's membrane. Okay, now outside what is there? Outside you have this two types of trophoblast. Okay, so in the trophoblast you have inner layer which is a cytotrophoblast. Okay, the outer layer, these are the mononucleated cells. Okay, and they undergo mitotic division. This is the outer trophoblast, which is called as syncytiotrophoblast. Okay, and these are multinucleated cells. They are not mitotically active. Okay, so this is what we have learned up till now. So two cells, two cavities, two types of trophoblast, week of two. Okay, so these are the three things which you should remember once we are discussing about second week. Two cells, two cavities, two types of trophoblast. Okay, so now we know that this is the cytotrophoblast, this is the syncytiotrophoblast. What happens further in this syncytiotrophoblast, especially where this inner cell mass lies, to that side the small lacunae will develop formation. Okay, this lacunae will get formed into the this syncytiotrophoblast. Okay, so that is called as lacunar stage. We'll go into the details. So 8th day we have already finished. Then what happens in the 9th day? The blastocyst is more deeply embedded into the endometrium and the fibrin coagulum is formed. The trophoblast development, particularly at the end of the embryonic pole, there are vacuoles which appear in the syncytium. These vacuoles fuse and lacunae formation. This is called as lacunar stage. Okay, these are the lacunae which are happening into the syncytiotrophoblast, and that is called as the lacunar stage of the blastocyst. Okay, now what is happening here in the embryonic pole? Flattened cells of the exocoelomic membrane originate from the hypoblast. This is the user's membrane. Okay, these are the flat hypoblast. These flat hypoblast will give rise to another layer which will line this particular cavity. So this is the flattened cells, exocoelomic user's membrane originate from the hypoblast. Okay, so you have the exocoelomic cavity, that is the primitive yolk sac covered with by the hypoblast plus extra embryonic membrane. Okay, so you have this yolk sac, primitive yolk sac, which is formed from the this hypoblast and the user's membrane. Okay, so this is the primitive yolk sac. This is the amniotic cavity. Okay, so clear up to this much. So 9th day. Okay, now what happens further around 11th to 12th day? Okay, now we have seen these are the hypoblast. This is the user's membrane. Now further, this is the cytotrophoblast and this is the syncytiotrophoblast. So in bit something new which is happening. Now let us see what is this happening. Okay, so blastocyst is completely embedded in the endometrial stroma by this time. There is a slight protrusion into the lumen of the uterus. There are sinusoids or the maternal capillaries which are congested and dilated. Then there is a establishment of the uteroplacental circulation. Maternal blood flows begins to flow through the trophoblastic system. The extra embryonic mesoderm, a new population of the cells, appear between the inner surface of the cytotrophoblast and the outer surface of the extra exocoelomic cavity derived from the yolk sac cells. Okay, so this is the trophoblast, cytotrophoblast, and this is the exocoelomic membrane. Okay, so between this exocoelomic membrane and this cytotrophoblast, another special type of cells which appear. Okay, this is nothing but the extra embryonic mesoderm. Okay, so this is the extra embryonic mesoderm. This you see is the extra embryonic mesoderm. Okay, sorry. Okay, so the extra embryonic mesoderm, a new population of the cells appear between the inner surface of the, so inside of the cytotrophoblast and outer surface of the exocoelomic cavity, you get a new type of cells forming the extra embryonic mesoderm. Okay, now what happens in this extra embryonic the mesoderm, the extraembryonic cavity that is called as chorionic cavity. You see small spaces which are happening inside the mesoderm. So these lacunae will fuse and a new cavity will get formed. Okay, so this extraembryonic mesoderm will have multiple cavities or the lacunae, and they will unite to form a big cavity. Okay, so that is called as chorionic cavity. So here you will have another cavity called as chorionic cavity. So up till now you have seen here is an amniotic cavity, here is a primitive yolk sac, then there is a development of the extraembryonic mesoderm. So in the extraembryonic mesoderm also you are getting a big cavity completely lined by all over except at one stage, this where this inner cell mass will be connected to the cytotrophoblast, that is called as connecting stalk. We will see that in the diagrammatic form in the next slide. Okay, so I hope you are getting all this layer by layer how it is getting formed. Okay, so large cavities develop in the extraembryonic mesoderm. Extraembryonic cavity or the chorionic cavity surrounds the primitive yolk sac and the amniotic cavity except where the germ disc is connected to the trophoblast by the connecting stalk. Okay, so now you have the extraembryonic somatic mesoderm, the extraembryonic mesoderm lying the trophoblast and the amnion. So this mesoderm, extraembryonic mesoderm, now gets split into two. Okay, so this is a somatic mesoderm and another is a splanchopleuric mesoderm. Somatic and splanchnic. So these are the one which lies the yolk sac is the splanchnic mesoderm, and rest of the mesoderm is the somatopleuric mesoderm. Okay, so the extraembryonic mesoderm also gets divided into two by the formation of this cavity. The one which lies lining the yolk sac is called as splanchopleuric mesoderm, and the one which the rest of the mesoderm you can say is a somatopleuric mesoderm. Okay, so the extraembryonic somatic mesoderm, the extraembryonic mesoderm lining the cytotrophoblast and the amnion is called as extraembryonic somatic mesoderm, and the extraembryonic splanchnic mesoderm, the mesoderm which lines the or the covers the yolk sac is called as extraembryonic splanchnic mesoderm. Okay, then there is a decidual reaction. Okay, cells of the endometrium will become polyhedral, loaded with glycogen and lipids. There are intercellular spaces which are filled with extravasate, and the tissue becomes edematous. Okay, so one important thing here you note, when the chorionic cavity forms, the extraembryonic mesoderm will get split into two, an outer somatopleuric layer lining the trophoblast and covering the amnion, and the inner splanchopleuric layer covering the yolk sac. So this part of the mesoderm, extraembryonic mesoderm, which covers the yolk sac, will form the splanchopleuric mesoderm. While outer somatopleuric, so you have outer and the one which lines this amnion, and rest is the somatopleuric mesoderm. Okay, so we will further see in the next few slides. Okay, so this is the chorionic cavity. Okay, so initially these are the small spaces which happen. Okay, now they are united and they will form a big cavity which is called as chorionic cavity. Okay, that also we have discussed. Now another thing which is happening here, the small part of the yolk sac gets split. Okay, so this is the yolk sac, a small part of it will get separated from it, and here they will form a exocoelomic cyst, and that time we are going to call this as a secondary yolk sac. So this primary yolk sac, what primitive yolk sac or the primary yolk sac, what we have seen here, initially it was a primary yolk sac, then there is a chorionic cavity is getting formed. Once this chorionic cavity becomes big, this cavity is surrounding this blastocyst, developing blastocyst all around except at a place which is called as connecting stalk. So connecting stalk is a site where the umbilical cord will get formed. Okay, now this by this time this is a primitive yolk sac or the primary yolk sac. After this chorionic cavity gets formed, a small part of this yolk sac will get pinched off and will form an exocoelomic cyst, and this primary yolk sac will be labeled as a secondary yolk sac now. Okay, so on 13th day the surface of the endometrium is healed. Okay, see the surface is healed. Okay, the occasional bleeding occurs at the implantation, and the blood flows into the lacunar spaces. There is a formation of the villus structure. Okay, now if you notice here, the villi are getting formed. Okay, so this is the cytotrophoblast and this is the syncytiotrophoblast, and this trophoblast is showing these projections. Okay, so you have the cytotrophoblast and you have the syncytiotrophoblast. These are the villi formation. Okay, so there is a formation of the villus structure by around day 13 or 14. Then it has got a cytotrophoblast and the syncytiotrophoblast. The primary villi will begin form. So these villi also you should know because they are asking the questions on it. We have this SBS at the end. Okay, so then there is a secondary or definitive yolk sac formation. Okay, so hypoblast produce additional cell inside the exocoelomic membrane during the second week. The primary or the primitive yolk sac is partially constricted and replaced by the smaller secondary or the definitive yolk sac due to proliferation of the extraembryonic mesoderm and remodeling of the exocoelomic membrane. Now, when this initially it was a primary yolk sac. As the chorionic cavity develops, a small part gets pinched off, and this primary yolk sac becomes the secondary yolk sac or the definitive yolk sac formation. And this hypoblast produces additional cell inside the exocoelomic membrane during the second week. The primary yolk sac is partially constricted and replaced by the smaller or definitive yolk sac due to proliferation of the extraembryonic mesoderm and the remodeling of the exocoelomic membrane. Now you have got this exocoelomic cyst. When a small portion of the primary or the primitive yolk sac gets pinched off during the formation of the secondary yolk sac in the second week of development, this detached remnant lies within the chorionic cavity as a cystic structure. Okay, so small part of this yolk sac will get pinched off and will lie into the chorionic cavity as a cystic structure. The chorionic cavity is the extraembryonic coelom that will expand. Okay, and there is a connecting stalk where the umbilical cord will get formed. This is the connecting stalk. So where the umbilical cord formation will happen. Okay. So what is the role of syncytiotrophoblast? The hormone production SCG will be produced. So by the end of the second week, sufficient quantities of beta SCG is produced, which can be detected by radioimmunoassay. 50% of the implanting embryos genomes are derived from the father. Okay. Now these genomes are foreign body material. Okay, so the body, so maternal body actually should reject this foreign material, but actually this is not happening. So 50% of the implanting embryos genome are derived from the father, which is a foreign body for the maternal immune system, and ideally it should be rejected like a transplanted organ. But why the immune system is not rejecting or the developing embryo, that is not very much understood. But it appears that one thing which you remember now, there is a shift from cell-mediated response to the humoral immune response, that is the antibody-mediated immune response, which protects the embryo from the rejection. So the one of the theory is that there is a shift from the cell-mediated immune response to the antibody-mediated immune response with the pregnancy status. Okay, so that is why the autoimmune diseases, okay, in which the cell-mediated response is predominant, they will have the favorable outcome. Okay, so manifestations of the immune disorders may change during the pregnancy, such as rheumatoid arthritis, multiple sclerosis, where the cell-mediated immune response predominates, will show the improvement during the pregnancy because there is a shift from the cell-mediated immune response to the antibody-mediated immune response, while the SLE, where the humoral immune response predominant, will show the affection of the pregnant women. So SLE will deteriorate while rheumatoid arthritis, multiple sclerosis will improve during the pregnancy. So if these diseases, the pregnancy happens during the pregnancy, the rheumatoid arthritis, multiple sclerosis, they will show the better outcome or the improvement in the condition. Okay, because of the shift in the immune system, while the SLE will deteriorate. Okay, so the abnormal placentation, normally it is generally either anterior or posterior wall of the uterus. Generally, it is the posterior wall upper part. Okay, so if it happens over the internal os, it will lead to the placenta previa, or if it happens in the tube, it will lead to the ectopic pregnancy. Okay, so the same thing what we have discussed, we'll learn in a diagrammatic way. Okay, we have seen that these are the upper tall columnar layer epiblast, and these are the flat cells. These are the hypoblast. Okay, this we have seen in the bilaminar gel disc. Then we have the amniotic cavity here, and then you have the primary yolk sac here. Okay, and this is the amnion which is there, and this is the Husserl's membrane. From the hypoblast, a layer happens which is the Husserl's membrane, and after that a layer, that is the extraembryonic mesoderm, gets formed. This is the primary mesoderm. Okay, so this is the extraembryonic mesoderm. Okay, there is another mesoderm that we are going to see, but that is the embryonic mesoderm, that is different. So please do not get confused. Okay, so this extraembryonic mesoderm is getting formed. Now we have seen the cavity is getting developed into the extraembryonic mesoderm, and this extraembryonic mesoderm will have now two labeled into two parts. One, the one which lies around the secondary yolk sac, will call as plankopluric mesoderm, while the rest of the mesoderm will be called as the somatopluric mesoderm. Okay, and this is the connecting stalk where the chorionic cavity does not develop. So chorionic cavity will surround everywhere this amniotic cavity as well as secondary yolk sac, except at one place where there is a connecting stalk. Okay, and this will give rise to the future umbilical cord. So you have these. So you will have this trophoblast. Okay, then you have this amnion. Okay, so this is the amniogenic layer plus somatopluric layer of the extraembryonic mesoderm. Then you have this chorion, that is trophoblast plus somatopluric layer of the extraembryonic mesoderm. Okay, so this is the somatopluric mesoderm, and this, the one which lies the yolk sac, is the splankopluric mesoderm. So this somatopluric mesoderm plus this amniogenic layer will form the amnion. This somatopluric mesoderm plus trophoblast, okay, will form the choria. Okay, so somatopluric layer of the extraembryonic mesoderm and the trophoblast will form the chorion. Then you have this extraembryonic coelom, also called as chorionic cavity. This is the splankopluric layer of the extraembryonic mesoderm, and this is the somatopluric layer of the extraembryonic mesoderm. Okay, so amnion, it covers the amniotic cavity and is contributed by the amniogenic cells from trophoblast and the somatopluric layer of the extraembryonic mesoderm. Chorion, it is contributed by the trophoblast, that is the syncytiotrophoblast plus somatopluric layer of the extraembryonic mesoderm. Okay, so the trophoblast divides into cytotrophoblast and the syncytiotrophoblast and will form the placental membrane. Okay, okay, so let us talk something about the yolk sac. Okay, so we have seen that this is the yolk sac. We have initially it was a primary yolk sac, then there was a secondary yolk sac, then there is a definitive yolk sac. Okay, so or also called as tertiary yolk sac. So these are the different types of the yolk sac. The same yolk sac gets further developed. So yolk sac is a membranous sac lined by the embryo called the yolk sac. Is made up of cells from the hypoblast layer of the bilaminar embryonic disc. So a membranous sac linked to an embryo called the yolk sac is made up of cells from the hypoblast layer of the bilaminar germ disc. So we have seen that there are two cells here, the epiblast and the hypoblast. From this hypoblast, you are getting this membranous sac. Okay, the sac is far more common name, but other name for the yolk sac is the umbilical vessel. Okay, this is also called as umbilical vessel. The yolk sac plays a crucial role in the early blood supply of the human embryos during the fourth week of embryonic development. A large portion of it is integrated into the primitive gut. So once there is a folding of this bilaminar or trilaminar germ disc, at that time this yolk sac will get incorporated into the embryo. So this actually, this yolk sac doesn't get part in formation of the embryo. Only the part which gets invaginated into the developing embryo. Okay, when there is a folding, the part of this yolk sac will become the part of the developing embryo. That will lead to the formation of the primitive gut. Okay, so that part will form the primitive gut. Okay, while the rest of the yolk sac will not take part into the formation of the embryo. And this yolk sac is connected to the developing primitive gut through a duct which is called as a vitalo-intestinal duct. All these are SBS for you. Okay, so okay, the allantois is also a projection from this secondary yolk sac. Okay, so this allantois is a projection. Okay, this is also an SBA. We will see. Okay, so by day 8 and day 9, you have got a primary yolk sac. By day 12 to day 13, you get the secondary yolk sac. Day 16, the allantois will appear. Week 3 is the hematopoiesis begins, and week 4 there is a folding and the primitive gut formation. Okay, so primary yolk sac is transient, secondary yolk sac is functional. Allantois arises from yolk sac endoderm. Okay, so the yolk sac endoderm will give rise to allantois. Okay, this yolk sac endoderm will give rise to allantois. The primitive gut is formed by incorporation of the yolk sac during the folding. As I already mentioned, as this developing embryo gets folded, this yolk sac will get incorporated into the developing embryo and will form the primitive gut. And the connecting between the two is the vitalointestinal duct. And this vitalointestinal duct failure of the closure will lead to the Meckel's diverticulum. This is also a question which is asked in the examination. Okay, so let us see here. You have got an amniotic cavity. You have got the primitive yolk sac, and you have got the Husser's membrane formed by the flattened endodermal cells. Okay, now further what happens, this is the chorionic cavity which is getting developed. Okay, this is the chorionic cavity, or the extraembryonic coelom is getting developed. So you have got a trophoblast here. This is the extraembryonic coelom. Then you have got the primary yolk sac will become the secondary yolk sac. Okay, then there is a folding. With the folding, what happens, this amniotic cavity will be like this here. Then this secondary yolk sac will get incorporated here and will form the primitive gut. There is a projection which is happening from this secondary yolk sac is called as allantois. Then there is a connection between the two is called as vitalointestinal duct, and this secondary yolk sac will be labeled now as a tertiary yolk sac. Okay, that is a portion of the secondary yolk sac not taken inside the body of the embryo, and here is the placenta which is getting formed. Okay, so this is actually the changes which are happening in the second week. The another change which begins is the formation of the primary villi. However, it will further go into the third week also. Now, what happens in the second week in the villi formation also begins by around day 12. Okay, so here also we have seen here, these are the cytotropoblastic cells and these are the syncytiotrophoblast. And you see these projections. These projections are nothing but the primary villi. You have a cytotrophoblast inside and covered by the syncytiotrophoblast. Okay, so this outer dark green is the syncytiotrophoblast, and these are the spaces filled with the maternal blood. Okay, this is the inside is the cytotrophoblast. The pale green color is the cytotrophoblast. Okay, so these are the primary villi. Okay, so in the primary villi, the outer is the syncytiotrophoblast, and outer layer is the syncytiotrophoblast, and the inner layer is the cytotrophoblast. So if you have the got a cut section, you will see that inside is the cytotrophoblast and outside is the syncytiotrophoblast. The core of the cytotrophoblast cells covered by the layer of the syncytium, that is syncytiotrophoblast. Okay, now by second it gets converted into secondary villi by day 13 to 15. You have got a cytotrophoblast core which is invaded by the extraembryonic coelom. Okay, so we have seen that the extraembryonic coelom is getting formed. Okay, now you are getting this embryonic mesoderm. Sorry, extraembryonic mesoderm is getting formed. So cytotrophoblast core invaded by the extraembryonic mesoderm. So you have got this green layer that is the extraembryonic mesoderm. Okay, now this fetal blood vessels will appear into this extraembryonic mesoderm that will lead to the conversion to the tertiary villi by day 17 to day 21 by in the week 3. Okay, so they are asking this development of this primary villi, secondary villi, or the tertiary villi. They may ask you the layers. Okay, the primary villi will have a cytotrophoblast inside, then the syncytiotrophoblast outside. So they can give you the syncytiotrophoblast inside, cytotrophoblast outside. They may give you that extraembryonic mesoderm outside, and multiple combinations they will give you, and will ask you the histopathological structure of primary villi, secondary villi, and the tertiary villi. So you should know what is happening. Here it is a cytotrophoblast covered by the syncytiotrophoblast. Here the extraembryonic mesoderm happens in the core, followed by the cytotrophoblast, followed by the syncytiotrophoblast. And in the tertiary villi, the blood vessels will appear. So in the center, you will have the developing blood vessel, followed by the extraembryonic mesoderm, followed by the cytotrophoblast, followed by the syncytiotrophoblast. cytotrophoblast. Okay, so primary villus by day 12, you have a core of cytotrophoblast cells covered by the syncytiotrophoblast. You have secondary villus by day 13 to day 15. Cytotrophoblast layer is invaded by the extraembryonic mesoderm, and tertiary villus will have the fetal blood vessels that invades the mesoderm. Okay, so that finishes the development of the second week okay, of the embryo. Thank you.